Imagine stepping into the future where the smartest learning machine isn't running Windows or Linux, but it's actually bubbling quietly in a test tube.
Right.
What if the next great supercomputer wasn't some giant server farm guzzling electricity, but a tiny vial filled with liquid?
It sounds like science fiction, but it's becoming reality.
We are talking about real artificial intelligence, but not in silicon chips. It's running purely on, well, on DNA molecules.
And that shift is potentially revolutionary. We're diving into some fascinating research published in Nature.
Okay, big deal journal.
Definitely. So from a team at Caltech, Kevin M. Cherry and Lulu Keen. And they've really kind of shattered expectations about where computation can even happen.
So our mission today is a deep dive into their paper, Supervised Learning in DNA Neural Networks. We want to unpack how they actually built a synthetic DNA circuit that can learn autonomously. And this isn't just, pre-programmed number crunching. This is a physical system forming actual chemical memory, molecular memory.
Exactly. And that concept of learning is the critical leap here. I mean, for any living thing, learning is just fundamental for survival.
Right? Sure, yeah. Like our brains or even our immune system.
Right. Your brain uses synaptic plasticity. Your immune system remembers pathogens it's seen before. The huge challenge for scientists has always been, how do you give something non-living, something not biological, that same kind of adaptive, complex ability?
Okay, so what was the roadblock before? People have tried building things with DNA circuits, haven't they?
They have, but previous attempts at molecular neural computation using nucleic acids or protein circuits, they often hit this massive limitation.
Which was?
The learning part, the optimization, figuring out the connections, the weights, that all had to happen in silico.
Meaning on a regular computer.
Exactly. On a standard computer. You basically used your laptop to figure out the brain, and then you manufactured a fixed hardwired DNA circuit based on those calculations.
So it was smart, but kind of static, like a fancy calculator that could only do one specific thing it was built for.
Precisely. It couldn't adapt or learn anything new on its own once it was made.
Okay, so if I'm tracking this, the laptop's gone now. We're saying the intelligence itself, the ability to adapt and change, is happening purely through chemistry, in vitro, in the liquid.
That's the breakthrough. The Caltech team demonstrated that the liquid chemical system itself autonomously forms and adjusts its own memories. It does this in response to molecular examples it's shown.
Wow.
Yeah, it acts like a true adaptive memory device, learning on its own terms, chemically. It feels like the major conceptual jump here. Computation moving from electrical signals to dynamic chemical concentrations.
But the authors set a really high bar for themselves, didn't they? They weren't just aiming for a simple reaction. They wanted something that could really generalize knowledge, like a student learning from a teacher.
They did. They set a high standard. They imagined an artificial cell observing a teacher's behavior and learning from multiple examples to generalize. And to really claim success, they laid out 4 key requirements.
Oh, what are they?
First, independence. This means the training data and the test data have to speak the same molecular language. No outside computer translated needed. It all happens in the same chemical pod.
Makes sense. And second?
Integration. This is crucial. It has to remember new examples without overwriting or forgetting the old ones. You can't learn A and then totally forget A when you start learning B. The memories have to build up.
All right, cumulative learning. What else?
Third is generality. It needs to use what it learned to process new data, even if that data is a bit noisy or corrupted or just slightly different from what it trained on. That's vital for any real-world use.
Absolutely. Real data is messy.
Exactly. And finally, stability. These chemical memories, they have to last. We're talking days, maybe even months after the training is done. Otherwise, what's the point?
That whole process, observing, integrating, strengthening connections, it sounds really familiar. Are we talking about something analogous to how our own brains learn?
Conceptually, yes, there are strong parallels. The way they train the system, it strongly resembles learning by memorizing. It's kind of like the statistical method. K means clustering, where you group similar data points together.
Okay.
But maybe the most exciting parallel is to Hebbian learning.
Ah, the classic neuroscience idea.
Yeah.
Cells that fire together, wire together, or neurons, rather.
Exactly that principle, but applied chemically. In this system, the input pattern, the data molecule, and the correct class label, the teacher signal molecule, are introduced at the same time.
So they fire together chemically.
Right. Their simultaneous presence chemically strengthens the pathway connecting them. That co-occurrence literally causes the concentration of a specific memory molecule to increase. That's the memory trace getting stronger. That's how the wiring happens in a test tube.
Okay, this is fascinating. Let's get down to the nuts and bolts then. How does this chemical learning actually work without tiny transistors or wires? What's the core mechanism?
The whole thing runs on highly specific chemical reactions called DNA strand displacement. This is the engine under the hood. It's how DNA molecules can be engineered to perform logic, like AND gates or OR gates, but purely chemically.
Can you give us an analogy for that? DNA strand displacement, what's that like?
Okay, think of it like molecular Velcro, or maybe a zipper. A DNA double helix is like a closed zipper.
Right.
Strand displacement happens when a new single strand of DNA comes along. That's our input signal. If it has a sequence that perfectly matches one side of the existing zipper, it can bind there.
And it pries the original strand off.
Exactly. It binds strongly and literally peels off or displaces the original complementary strand. This changes the structure, and crucially, it can release a new chemical molecule that acts as the output signal for the next step in the computation.
Incredible. It's like a tiny specific chemical chain reaction.
Precisely. Highly programmable, very specific interactions.
Okay, so let's picture the memory device again. It starts out blank, right?
Yeah.
All the molecular memories, the weights, as they call them in machine learning, are at 0.
Correct. A blank slate, chemically speaking. Then comes the training phase. The researchers introduced the molecular input patterns, that's the data alongside the class labels, which act as the teacher.
Okay.
These molecules then chemically interact, and they collectively activate specific components within the solution. This process directly adds the input information to the memory associated with that specific class label.
And the memory itself, the weight, isn't stored as like a digital bit, 0 or 1. It's stored as a concentration of a molecule.
Exactly right. If the connection between a specific input pattern and its correct label is strong, meaning it's shown repeatedly, the system literally produces more of a specific chemical marker. They call these activator molecules.
So higher concentration means stronger memory.
Yep. The concentration of that activator molecule becomes the chemical memory token. More molecules means a stronger weight for that pathway.
But wait, how does the system then use those weights to classify something new? You mentioned calculating a weighted sum. How does a liquid, like, compute a sum? That sounds weird. Yeah, it's not intuitive like silicon math, but it's elegant chemically. Those active memory tokens, the activator molecules that are now present in higher concentrations for learned patterns, they connect to a separate molecular processor circuit.
Okay.
When a new unknown test input molecule is introduced, it chemically interacts with all those learned weights simultaneously. The magic is that the resulting output chemical signal from the processor is proportional to this weighted sum.
Meaning the memories with higher concentrations contribute more strongly to the final output signal.
You got it. More concentrated memories push the reaction further or faster, leading to a stronger output signal for that particular class.
Ah, okay. So the output signal strength reflects the sum. And that leads to the decision-making part, the winner-take-all competition. Absolutely. The system is designed so that the output pathway, yielding the largest chemical sum, meaning the input most closely matches that strongly reinforced memory pathway, gets dramatically amplified.
So it boosts the strongest signal and suppresses the others.
Right. It acts as the decision maker, producing a clear, unmistakable on or off signal to classify the input. It's how the system definitively says, okay, based on what I've learned, this new pattern looks most like Class A.
And they were careful about the training data too, right? They didn't just throw random examples at it. They used a good teacher strategy.
Yes, that's important. Just like you wouldn't want a student learning from bad examples, they developed a chemical filtering strategy. They essentially ranked the training patterns and selected only the high-quality, above-average ones.
So they made sure the DNA network learned from the best representations of the data first to set those chemical memory concentrations accurately.
Exactly. Better training data leads to better learning and classification.
Okay, this setup sounds incredibly sophisticated. It brings us to the real proof of concept.
Yeah.
What was the big test? What did they actually ask this DNA brain to do?
Well, they gave it a classic challenge, something that's basically a rite of passage for pretty much every digital machine learning algorithm out there. classifying handwritten digits.
No way. You mean like the amnested database stuff, those little black and white images of digits that humans wrote?
The very same. Those scratchy, slightly imperfect, 100-pixel, or in this case, 100-bit representations of digits like 0, 1, 3, 4, 6, and 7.
And the DNA network could learn to tell them apart.
It could. They successfully trained different versions of the network to classify 3 distinct pairs of these 100-bit digits.
What?
For instance, telling a 0 from a 1 or a 3 from a 4.
Just think about that. 100 different chemical inputs needed just to represent one handwritten digit. That's complex.
It is. And here's where the scale gets really kind of mind-bending. To handle that 100-bit input and maintain just 2 separate memories, say one for the average Jerome and one for the average 1, this network involved over 700 distinct species of molecules.
700. Swirling together in one test tube.
In a single test tube, and across the entire process, the learning phase and the testing phase, they designed and used more than 1,200 unique DNA strands.
Wow. That level of molecular orchestration in just, a drop of liquid is staggering to think about.
It really is. And the system proved it wasn't just theoretical, it was robust. It met all those key requirements they set out.
Like the integration part. Did it remember sequences?
It did. They showed it successfully absorbed training data presented in different batches and different orders without the later training messing up the earlier memory.
And generality. Could it handle imperfect data?
Yep. Powerfully so. It accurately classified new test patterns even when they were deliberately made noisy or corrupted chemically. That showed that wasn't just memorizing, it was generalizing.
That's huge for real-world potential. What about stability? Did the memories last?
They did. The researchers checked, and the molecular memories were stable. They could still perform accurate classification days after the initial training was finished. The chemical weights held.
Okay, and you mentioned something about impurities. That sounds important for actually making these things.
Yeah, this was fascinating from an engineering standpoint. Apart from some specific reporter molecules, the entire system, all those hundreds of DNA strands, was built using mostly unpurified DNA.
Unpurified. So like kind of messy, chemically speaking?
Relatively speaking, yes. It suggests the system has this inherent robustness. It's highly tolerant of molecular imperfections and chemical noise, which is actually a major potential advantage over electronic circuits that need extreme precision and purity.
Okay, so we've established that liquid chemistry, DNA to soup, can actually learn complex tasks autonomously. This brings us to the so what? What does this mean for us, for the real world? Where could this embedded intelligence go?
Well, it immediately opens doors for smart decision-making systems that can operate in places where normal electronics just can't.
Like inside the human body. Is that the big dream here?
That's definitely a major potential area. Think about biomedicine. We already use simple DNA circuits for some diagnostics, but if they can learn...
Then they could become adaptive therapeutics.
Exactly. Imagine a smart drug delivery system that learns. It could remember past encounters with specific disease biomarkers, like a cancer signature.
And prove its response over time based on how often it sees that signature.
Precisely. A therapeutic that adapts to the patient's specific disease progression dynamically.
That's incredible. What about other areas? Materials?
Soft materials, definitely. DNA circuits are already being used to control the physical properties of materials, making them expand or contract, for example.
So materials that can change shape on command.
Right. Now, add learning. Imagine materials that can adapt their response based on past experiences. A material that learns to react differently to pressure over time, or learns to adapt to changing temperatures or chemical environments.
Like a self-healing material that gets better at predicting where damage might occur. Adaptive soft robotics.
That's the kind of direction this opens up. But the researchers, Cherry and Kian, they're also very clear about the next big hurdles.
It's not ready for prime time just yet.
No, there are major challenges. One huge goal is unsupervised learning. Right now, the system needs that teacher signal the labeled examples.
But the ultimate goal is for it to learn just by being exposed to its environment, training itself.
Yes. Enhancing its own capabilities through unguided exposure to its molecular world. That's a whole other level of complexity.
What else is limiting it?
A big practical one is reusability. At the moment, these DNA neural networks are basically use-once systems. The computation uses up the stored chemical energy, driving the system towards equilibrium, like a battery running down.
So you can't just reset it and teach it something new easily.
Not with the current designs. To really scale this up for complex, ongoing tasks, we need to develop sustainable, reusable molecular computation. Systems that can be reset and learn again and again.
And getting more complex probably means getting more organized spatially, not just a soup.
That seems to be the consensus. To dramatically increase the complexity, scientists need to introduce spatial organization. They're looking at things like using phase-separated DNA condensates.
Which are like tiny, self-contained droplets.
Exactly. Microscopic droplets that could act as organized compartments within the solution. This kind of spatial architecture could potentially scale up the network complexity by an order of magnitude, maybe more, moving towards genuinely sophisticated molecular thinking machines.
Wow. So this research really does shift how we think about intelligence, doesn't it? Suggesting that adaptive, decision-making abilities aren't just for biology or electronics, but can be engineered right into non-living matter. Let's just make sure everyone listening has the key takeaways locked in from this deep dive.
Okay, first, the big one. DNA molecules can autonomously perform sophisticated supervised learning. They can classify patterns entirely in vitro in the test tube. This moves way beyond older, fixed, hardwired chemical calculators.
Right. Second, the sheer scale of the engineering is incredible. They demonstrated this with a 100-bit input to memory network that needed over 700 different types of molecules working together in sync in one solution.
Yeah, the complexity is amazing. And third, this system is surprisingly robust. It showed those critical learning characteristics, integrating new data, handling noisy patterns, and it did so with a remarkable tolerance for chemical impurities.
It really does start to blur the line between what we think of as a living organism and a computational device. It suggests the architecture of intelligence might be way more flexible than we thought. Maybe intelligence can live in chemical concentrations, not just electrical flows.
It opens up fundamental questions. And that leads to the really provocative closing thought. If these DNA circuits can learn so well when given a teacher, what happens if we take the teacher away?
You mean just let them learn from the environment, unsupervised?
Exactly. What kind of spontaneous, unsupervised intelligence might actually emerge when we just unleash these complex molecular systems to learn entirely on their own in a test tube? What would that even look like?